Thank you, Shivali, for the introduction, and I would like to thank BSI committee for letting me present my work here on 2024 conference. So my name is Akansha Singh. Currently, I'm a research assistant professor at UT Southwestern Medical Center, but the work I'm presenting today, I have done this when I was part of Oklahoma State University. So today I'm going to talk about adrenergic receptor signaling and its regulation on CD40 agonist antibody mediated anti-tumor immunity. So CD40 receptors, they are TNF receptor family member, and their expression is found in many APCs like dendritic cells, macrophages, B cells, and other non-immune cells like platelets, your epithelial cells and endothelial cells. So the agonist CD40, which is designed to mimic the CD40 ligand when it interacts on APCs, and here we're going to talk more on the dendritic cell side. So when they interact on the CD40 receptor on dendritic cells, it enhances the production and the expression of antigen-presenting molecules like MHC and other co-stimulatory molecules like CD86 or CD80 on these cells, which further enhance the priming of T cells by these mature dendritic cells. But when this therapy in preclinical model, it showed wonderful results for many tumor model, melanoma, colorectal cancer, and lung cancer, whatever you can think of. It worked very nicely in preclinical models, but when it was taken to the human trials, where humanized CD40 antibody were used, they showed very low efficacy, treatment efficacy, and that was associated with the very small half-life of these antibodies. Also, because CD40 is expressed by many other cell types, there was non-specific cell targeting and very high toxicity, because of which the lower dose was used in the clinic to avoid those toxicities, which further reduce the treatment efficacy of CD40 antibody and immune activation. So to mitigate this limitation, many groups are working in generating next-gen CD40 antibodies, but because this treatment still hold merits in activating dendritic cell mediated immune activation against tumor, so many groups are working on different aspects of this immunotherapy. Some are working on modifying the antibody design itself by clustering, by using FC engineered antibody to cluster more CD40 on dendritic cell, using bispecific antibodies to enhance the interaction of dendritic cell with other cells like tumor cells, T cells, and by modifying the approach of drug delivery, that is, instead of using IV injection, intratumoral injection, so that off-targeting activation and related toxicities can be reduced. But in our group, what we focused on was on the intrinsic factor in a human body that might be affecting the dendritic cell activation that is mediated by CD40 receptor activation. For that, we looked at the adrenergic receptor functioning, and we found that norepinephrine, which is released by the adrenergic neurons and its level is increased under stress condition, which is commonly seen in cancer patient, when it binds on the dendritic cell norepinephrine receptor, it decreases the cell efficacy to get primed, to get licensed or mature, and that might impact the CD40 mediated cell activation too. So why to select adrenergic receptor? So there are many clinical studies that are using beta blockers, that is beta adrenergic receptor inhibitors, by themselves or in combination with other surgery and other immunotherapies. So what it does in a immunologically cold tumor or in patients that shows chemoresistance or resistance to the current treatment, the population of cytotoxic cells that are capable of killing the tumor cells, it decreases. And the population of suppressor cells in tumor microenvironment that subsides the function of these cytotoxic cells enhances, which ultimately leads to tumor progression. And also it was found that activation of beta adrenergic receptor in cancer cell, when norepinephrine bind to the receptor on cancer cell, it increases their many apoptotic pathways, and it enhances the tumor chemoresistance or radio resistance capacities. By using beta blockers by itself, not in combination with other therapy, by itself decreases the suppressor T cell population, but increase in cytotoxic cells was not observed. But when it was it is given in with the combination of other immune therapies, very stark difference between the levels of these cytotoxic cells and tumor progression was seen. So this is already an FDA approved treatment that is in the patient, and we wanted to see whether it can be synergized with CD40 therapy. For that, what we did, we isolated dendritic cells from the bone marrow of mice, and then we differentiated them between naive and induced cells. So the induced cells were exposed to tumor cell lysate in vitro, and the naive cells were not exposed to any stimulation. So we wanted to see whether this adrenergic signaling is impacting the induced cells, or it works at the level of naive cells too, that are not exposed to any antigen. And then we treated these cells with beta adrenergic receptor agonist, that is isoproterenol, and in combination or without CD40 agonist antibody. And we incubated these cells for 48 hours, and then we looked at the activation status of these dendritic cells using flow cytometry, release of pro-inflammatory cytokines, expression of pro-inflammatory cytokine gene, and the NF-kappa B signaling molecules, because CD40 works through the CD40 receptor. When it activates the NF-kappa B pathway. So it is the major pathway through which it activates the dendritic cell response. So what we observed that either is the naive dendritic cell or induced dendritic cells, if isoproterenol was given, the beta adrenergic receptor was stimulated, there was a significant decrease in the surface expression of MHC2 molecules, CD86, and CD40 molecules on the cell surfaces. And when it was given in combination with alpha CD40, that is CD40 agonist antibody, the similar effect on naive cells as well as induced cells was observed. Then we looked at the NF-kappa B pathway molecule. So there was no direct indication in the literature how activation of beta adrenergic receptor might be influencing CD40 signaling, but based on the indirect evidences that is associated with G protein coupled receptor activation and the NF-kappa B pathway, how they might be interacting, we hypothesized that isoproterenol, when it interacted with beta adrenergic receptor on the cell surface, it will increase the cell levels of beta arrestin 2, which is known to inhibit the phosphorylation of I-kappa B alpha subunit of IKK complex. So phosphorylation of I-kappa B alpha is important for further slicing of NF-kappa B complex into activated form, and it is then transported to the nucleus of the cell. So we looked at the levels of phosphorylated and unphosphorylated I-kappa B alpha levels in the cells, and as we expected, the levels of phosphorylated I-kappa B was decreased when we stimulated the beta adrenergic receptor on these cells in absence as well as in presence of CD40 antibody. And this is the direct correlation we thought of that how this might be impacting the NF-kappa B pathway. And another way by which beta-adrenergic receptor might be impacting the NF-kappa B as mediated activation of gene is by increased level of CRAB in the nucleus, and it might compete directly with activated NF-kappa B complex for the gene binding site. And when we looked at again the phosphorylated and unphosphorylated form of CREB, and we observed that it was highly increased. Phosphorylation of CRB was highly increased with isoproterenol treatment, which is an agonist of beta-adrenergic receptor. Then we looked at the actual response, like if NF-kappa B pathway is activated, what is the resultant gene expression and the cytokine release that it's usually observed, whether there was impact there or not. So we looked at the gene expression of pro-inflammatory gene, cytokine gene like IL-1 beta, IL-6, and IL-10. And what we observed that the expression of all these all these cytokine gene was reduced. In the induced cells, we did not see any changes happening in the naive cells, maybe because of the lack of stimulation by in the dendritic cell microenvironment. But in the induced cells, we observed a significant decrease in these gene expression, as well as the release of the protein, that is the cytokine by itself in the culture supernatant. Then we wanted to look at the efficacy, like whether we can enhance the CD40 efficacy in the mice by using low doses of CD40 antibody and combining it with the beta blocker, that is propranolol. So propranolol, again, is the drug that is used in clinic and is FDA approved. And we combined it. We started the injection of propranolol after 5 days of tumor induction. We used MOC2 tumor model, that is head and neck tumor model, and it's highly immunosuppressive. It has highly immunosuppressive tumor microenvironment. That means it has a high concentration of MDSCs and M2 macrophages in the tumor microenvironment and lower infiltration of cytotoxic T cells. Then we, after starting daily propranolol injection of 200 microgram 5 days after the inoculation, on day 7 and 7 days later to that, we just gave 30 microgram of CD40 antibody intratumorally, and we looked at the tumor volume on the day 12 post-treatment, and we looked at the immune cell activation status through flow cytometry. So what we observed after the treatment, that propranolol by itself reduced the tumor burden, but the change in the tumor volume was significant when combination, whether combination treatment was given. And then we looked at the CD3 positive T cell population infiltrating the tumor. Again, like with CD40 BiTEs monotherapy and propranolol used as a monotherapy, there was a slight increase, but the change was non-significant. With the combination, we saw a significant change. And then we looked at the specific T cell number, that is CD8, which was again increased with the combination treatment, and the ratio of CD8, CD4 T cells was also higher. So why the ratio of CD8, CD4 T cell is important? Because it's one of the major T cell score that is observed in the clinic, and a higher CD8 to CD4 ratio score is considered good in the clinical parameters. And further, we looked at the activated CD8 T cells, that is granzyme B positive CD8 T cells, and the ratio of regulatory T cells, that is CD4 FOXP3, which are known to suppress or regulate the cytotoxic T cell function. And we observed that the CD40 monotherapy as well as the combination enhanced the ratio, although significantly, although the median was little different. So here you can see how the the treatment with a combination treatment enhanced the CD8 T cell population in the tumor microenvironment as in comparison to all other groups. And the granzyme B expression was also observed on those T cells. Now, next, we wanted to see how the dendritic cells are doing themselves. So again, with the treatment, monotherapy of propranolol and propranolol alpha CD40, we observed that CD3 T cell, CD11C positive cells were enhanced in tumor microenvironment, along with the expression of MHC2 and co-stimulatory molecules like CD86 and CD40 was higher. So with that, we conclude that using beta blocker can enhance CD40 mediated immune cell activation, even even at the suboptimal doses, and by enhancing the activation of dendritic cells, which further can activate naive T cells, increasing the infiltration of T cells in the tumor microenvironment and killing the tumor cells. So with this, I conclude my work presentation, and I would like to thank all my lab members and my faculty mentor, Dr. Ashish Ranjan. So one more thing I want to say about this treatment, that here we just looked at the one aspect of propranolol or beta-adrenergic receptor, but as we are working on the different treatment module of anti-tumor immunity and taking it to the clinic here at UT Southwestern, it holds a very promising results with other treatments, anti-tumor treatments, where a suppression of CD8 cytotoxic T cell is observed, and it should be pursued further. Thank you.
Absolutely. Thank you so much for your talk, Dr. Singh. This was very, very interesting to hear about, and we've got a full slate of questions for you as well.
Sure.
So I mean, perhaps before I jump into the questions that we have in the chat, I might ask a personal one. And that is that I'm always very interested in understanding how DCs are doing a better job of priming T cells to infiltrate the tumor microenvironment when there's all of these barriers surrounding it. And so I'm curious, in this particular situation where you're giving propanol and anti-CD40 stim, do you have any idea of like which mechanism of infiltration we're actually boosting here for T cells?
Yes. So because it's just 15-minute presentation, I had to rush through all the things. So we are giving intraperitoneal injection of propranolol, beta blocker. So we are not influencing the immune cells that are in the tumor, but the overall body of the mice. So what has been seen that propranolol injection influences dendritic cells at the level of their production in the bone marrow. So the cell, if when I remember a paper where they were looking at the arthritis model, and when they blocked the adrenergic receptor in the body, there was an enhanced population of dendritic cells. So first of all, propranolol is working at by increasing the number of dendritic cells in the body itself, and because of the signaling that was happening in the tumor microenvironment, dendritic cell goes to the tumor microenvironment, where because propranolol is modulating the suppressor cells in the tumor microenvironment, like working at the level of MDSCs and macrophages, there was an enhanced activation of dendritic cells that we are seeing, which then goes to tumor draining lymph node, and there they are activating CD8 T cells. So this activation of dendritic cells, or the maturation or licensing of dendritic cell, is happening in the tumor microenvironment, but the activation of CD8 T cells or priming of CD8 T cells happens in the tumor draining lymph node, and then they come back again to tumor microenvironment. So that's how the circle works. But propranolol acts at every level to with all the cells, and it increases T cell proliferation too if given systemically, and it increases the proliferation of dendritic cells too. So it overall increases the number of cells present in the body.
I see. And then there's sort of this homing driven or inflammation driven homing of things.
Yes.
Yeah. Very interesting. And you sort of alluded to this as you were talking about the MDSCs, but can you speak a little bit more to the impact of Tregs in the tumor microenvironment in this case?
Yes. So Tregs are, because there could be T cells in the tumor microenvironment, but what kind of T cells are there is very important. So CD4 T cells, that are helper T cells, are required to activate CD8 T cells too. The reason we focused on dendritic cell because they are a very unique APC that can cross-present tumor antigen to CD8 T cells and directly activate CD8 T cells. But for the other cells to activate CD8 T cells, other APCs to work, they work through CD4. So if we have a cell which instead of activating CD8 T cells, they can suppress CD8 T cells because they interact with each other, CD4 and CD8 T cells. And when a cytotoxic T cell coming in the tumor microenvironment and they are Treg cells, instead of boosting their activity, they suppresses the activity of cytotoxic CD8 T cells, which results in, again, tumor growth and less activity of T cells in the tumor microenvironment.
Got it. That's wonderful. And I guess one last question that I'll ask. It's sort of a long one, but you talked about a lot of the cytokines in this case. Were there any other cytokines in the induced DCs beyond the ones that you've already described? And in that case, how do you think the induced dendritic cells were altered in the context of activation and function?
Yes, so for dendritic cells activation, I just selected these to see whether there's a change in cytokine production or not. But yeah, there are other studies where they have looked at the cytokines and the changes in the cells that is related to the migratory capacity of the dendritic cells, also antigen processing of these dendritic cells because those study was already there and they looked at the adrenergic receptor that are alpha type and beta 1 type and they are more associated with those. So in this study, I focused on beta 2 adrenergic receptor. So I looked at these cytokines which are like this receptor is known to influence these cytokines and I just wanted to look at the activation status mediated by CD4 activity. But yeah, like there are other studies where they looked at the different capacities of dendritic cells and how it is influenced by adrenergic receptor.
I see. All right. Well, honestly, this was so wonderful. I learned a lot from your talk. Thank you so much, Dr. Singh. It's truly been a pleasure hearing your work.
Thank you. And thank you again, BSI, for giving me the opportunity to speak here.